Hello, I'm Dr. Poirier and I'm the head of science at Vidiia. I lead the company's scientific strategy, specializing in developing new tests and products and making sure that they are rigorously validated. In a startup like Vidiia, being the head of science means wearing a lot of hats. In the lab, my job is to coordinate and prioritize the validation or improvement of our product and molecular assays. I always make sure that the science behind our product is solid, properly tested and proven. In diagnostics, people need to trust the results. Out of the lab, I explore emerging technologies and work closely with our product team to shape what we build next, so we keep up with the point-of-care market and its needs. I'm also in charge of explaining and communicating our science to partners, investors and the wider team, making sure that our work is understood and trusted. For me, it always comes back to the application. When using the VH platform, you are building a test for point of care, not a fully equipped lab. To build an assay, I start with identifying the target gene using genomics tool. Once the target is identified, I design LAMP primers. This is the main step that differs from PCR because LAMP uses multiple primers per target and the design is more complex. Once the primer set is designed, I validate them using our colorimetric lyophilized reagent, the VH-LyoMM. When paired with our AI LAMP technology embedded in the VH6 device, our reagents allow us to thoroughly validate the primer set and set the test parameters by analyzing amplification curve. The next steps will be to determine the sensitivity, specificity, cross-reactivity and limit of detection of the primers. PCR is the accuracy benchmark, but it relies on thermocyclers that tend to be expensive, power-hungry, and difficult, if not impossible, to make truly portable. It is therefore only for use in laboratory control environment. Our VH platform relies on LAMP assisted by AI, making it PCR grade whilst being truly portable and providing quicker results, making it applicable to near-patient and point-of-care. LAMP's biggest advantage is that it is isothermal, meaning that it runs at a single constant temperature, so there is no need for a thermocycler. That strips out the expensive, power-hungry instrument and lets users run molecular amplification on a compact, battery-powered device. It is also on average faster and more tolerant to inhibitors, making it compatible with rapid sample preparation, like for example our VH Rapid Prep. Finally, the results can be read by colorimetry, removing the need for expensive fluorescence optics. In the VH platform, we also coupled colorimetry with AI interpretation to build amplification curve and obtain quantitative data. Where PCR still wins is multiplexing. PCR can handle the amplification of multiple targets in one reaction more easily. Quantitative aspect of PCR using CT value has also been characterized across a wide dynamic range of samples. These features make PCR the regulatory gold standard being used in centralized laboratory with automated workflow available. AESI, standing for AI-enabled signal intelligence, is watching the changes in the LAMP reactions throughout the amplification. As nucleic acids are generated, the color and transparency of the LAMP reaction change, and the AESI continuously tracks how these changes develop over time. The AESI captures subtle change the eye would miss, and that is the AI in AI LAMP. And it is what lets a colorimetric reaction perform to the level of lab-based PCR. It is mostly because the detection mechanism is different. Fluorescence watches the DNA amplification directly and in real time, whilst colorimetric is indirect and senses a byproduct amplification. Our colorimetric LAMP reagent is metal ion based. In short, the DNA amplification generates some byproduct that will trigger the dye to change color. Classically, the color change is read by eye at the endpoint as a simple yes or no answer, which is subjective and can be difficult to read near the limit of detection. What changes for us is the eye. The AESI tracks the color and transparency of the lamp reaction over time and turns it into objective, time-resolved quantitative results rather than a binary one. The proof is in the parallel comparison on assays like COVID-19, where our colorimetric VH platform matched the limit of detection of a commercial fluorescent instrument. With AI and proper validation, colorimetric detection delivers a comparable performance at far lower cost and complexity, which is what a decentralized users actually need. Those numbers highlight two important things. First, the numbers themselves. 100% specificity means no false positive, which matters because it is a known misconception about LAMP. And 98% sensitivity against gold standard PCR means that we are matching PCR's accuracy in a portable format. Second, these numbers cleared regulatory approval during the COVID-19 pandemic, so it proves the capabilities of our platform. With PCR and most fluorescent systems, you need to develop in the lab and then have to re-engineer the assay onto a separate point-of-care platform. And that handover costs time, money, and sometimes performance. Our advantage is that the device that you develop is the device that you deploy on, with the same reagents and sample preparation. So you are validating in real-world conditions from day one. It is also cheaper and faster to iterate than PCR. The best fit is an assay developer, a research and development scientist, or a molecular biologist working on a target that is ultimately meant to leave the lab for point-of-care application, whether it is for human health, veterinary, agri-food, or environmental. It is especially interesting for people who want an open environment they can iterate on faster and cheaper than PCR. We are not building for large-scale and multiplex diagnostic. This is not us, and we don't pretend that we can compete there. The platform is for everyone else, for the researcher whose targets need to work outside a centralized lab. To me, it is a vet who gets a molecular-grade answer in under an hour and can decide there and then to isolate an animal instead of sending a sample off and waiting days. It is a nurse in a community clinic, a farm manager, a food producer testing on site. The point is that the person running the test does not need to be a molecular biologist and they don't need a lab behind them. Democratizing molecular testing means stripping out the things that kept it centralized, such as the cost, the cold chain, and the big instruments. In 2 to 3 years, I would most want to see it where waiting for a lab genuinely changes the outcome. And for me, that is a veterinary clinic. A vet who can confirm an infection on the spot can isolate or treat an animal the same day, instead of waiting days while it potentially spreads through the rest of the herd. And it is realistic, not a someday ambition, as we already have active development there. So it's why I think we can show real-world impact soonest, then build out from a proven case. I wish more people would understand that point-of-care diagnostic is not here to replace the central lab, but to complement it. There is a tendency to frame it lab versus field, but they do different jobs. The central lab is unmatched for complex, high-throughput, multiplex work and confirmatory testing. What point-of-care adds is speed at the place where the decision is made by giving a result in less than an hour, where it's needed. This results in early intervention and care instead of waiting days. The two work best together. The rapid testing at the front line to triage, isolate or treat immediately, with the lab behind for confirmation and behavior work. The win is not replacing the lab, it is closing the time gap between taking a sample and acting on it, which is often where outcomes are actually decided.